Hi there, my name is Draško, and I am a researcher at Ruđer Bošković Institute. Development of applications that might contribute to the creation of improved cancer drugs is the main goal of my research. Formerly, I was chief technologist at Hewlett-Packard Company and responsible for implementing and scaling NAMD and ANSYS on HP Helium public cloud. I did my postdoc research at Rijeka University, teaching students, administering Bura supercomputer, and developing Vini in silico model of cancer. My main awards are recognition by HP for significant innovation in the field of HPC cloud systems, membership in the Hewlett-Packard Hall of Fame. During the development of Vini, I realized my goal was aligned with that of DrugBank to help develop better drugs. This is the reason for my participation in today's DrugBank academic research webinar. Let's talk about Vini in silico model of cancer. Vini is a high-performance computing application that performs virtual drug screening for various diseases. There are many structure-based virtual drug screening programs. Some of them are commercial, while others are open-source applications. They all boast about being the top in their class. However, what makes Vini outstanding is the use of pathways that describe diseases. In silico modeling of cancer involves combining findings from biological literature with computer-based models of biological systems, and that in order to conduct investigation of hypotheses entirely in the computer laboratory. The pathways of cancer diseases are graphs presenting biological processes in cancer. Vini uses cancer pathways which are entirely built on the experimental facts found in the literature. Vini can also evaluate these hypotheses with the results obtained by experimental methods. Thus, it all makes the Vini in silico model of cancer an outstanding platform for the study of processes that contribute to the onset and development of cancer. What's the biological precedence for using pathway-based virtual drug screening? First, many virtual drug screening methods have been developed for identifying single target inhibitors based on the strategy one target, one drug. Hit rates of these methods are often low because they cannot capture features that play key roles in the biological functions of the target protein. Furthermore, single target inhibitors are often susceptible to drug resistance and are ineffective for complex diseases such as cancer is. Multi-target, multi-drug strategies can reduce the possibility of developing drug resistance. However, they ignore the effects of drugs on other, that means non-target proteins, which can then lead to worsening instead of improving the course of the disease. Therefore, it is necessary to take into account all receptors, that means proteins and substances and genes playing a role in the onset and development of diseases. But then, it is extremely important to know how these receptors interact with each other. Only pathways can provide this information. Because of that, Vini uses a pathway-based multi-drug strategy for virtual drug screening. Drug repurposing is the process of uncovering new indications of approved or failed abundant chemical compounds for use in a different disease. Drug discovery is the process of identifying chemical compounds that have the potential to become therapeutic agents. Vini is able to perform both drug repurposing and drug discovery. For drug repurposing, Vini uses drug structures from DrugBank. For drug discovery, it relies on the PubChem database. Why we use DrugBank? Because it is the largest database of drug structures that we are aware of, besides as academic users. And for this Vini development, we have successfully applied for free access to DrugBank resources. Let's take a look under the hood of Vini. Vini collects KEGG pathways from KEGG databank, drug and compound structures from DrugBank and PubChem, genomic and transcriptomic data from Cosmic Database, Protein Databank, and UniProt. Then it constructs data flow matrices, performs data flow optimization, and predicts drug candidates. This is a little more complex data flow schema. As you can see, there are pretty lot of program modules in Vini, and we will not go deep in this data flow, but only to mention that Vini has application interfaces to databases like UniProt, Cosmic, KEGG, etc. And also works with a program like Autodoc, Vina, like AlphaFold, and of course at the end, if it computes SLAM. Where to use and where not to use Vini. It is a structure-based virtual drug screening tool working on metabolic pathways of diseases. Therefore, it can be used for virtual drug screening. As it is also in silico model of cancer, it can be used for computer-based analysis of the main hallmarks of cancer and as a testbed for various cancer-related hypotheses. However, as it doesn't take into account unwanted side effects of drugs, their pharmacokinetics, for example, and pharmacodynamics, Vini is not a tool for predicting either drug efficacy nor drug efficiency. What are the basic premises of Vini? First, drug or chemical compound producing the low data flow speed in data flow graphs is likely a good candidate to initiate the slowdown of cancer disease or even stop it. Then, if a drug is a good candidate, then it deserves further in vitro and in vivo analysis. And if a chemical compound is a good candidate, then toxicity analysis must follow. Pathways. They are graphs that describe metabolic processes within organisms. Graph nodes represent the names of receptors, which could be either genes, proteins, or substances. Edges represent interactions between receptors. Some pathways are based on published experimental evidence, while others are computationally generated. Vini uses cake pathways based on published experimental evidence. There are thousands of cake pathways available. Vini is able to use all of them. Many of these pathways describe metabolic processes in human disease, including cancer. Here we can see some main hallmarks of cancer. For example, sustaining proliferative signaling, second evading growth subpressures, activating invasion and metastasis, enabling replicative immortality, inducing angiogenesis, and resisting cell death. But it is to point out that any cancer pathway must necessarily describe at least 6 basic mechanisms of cancers. Here we can see an example of one cancer pathway. This is a general cancer pathway. And the graph nodes are here in green, then hallmarks of cancer in gray, arcs connecting receptors. And what we need to go a little deeper in are eigenvalues, Markov chains, mixing rates of Markov chains, and data flow graphs to understand how Vini actually works. Eigenvalues are a special set of scalar values, and they are associated with a set of linear equations, most probably found in matrix equations. Eigenvalues of matrix A are scalars satisfying equation 1, which expanded leads to the characteristic polynomial in next equation numbered 2. The roots of this polynomial are eigenvalues, and we note them here, lambda. Mixing rates of Markov chains. Markov chain is a stochastic model describing the sequence of possible events in which the probability of each event depends only on the state attained in the previous event. Its transition matrix T describes the probabilities of particular transitions. In case of symmetric random walk, the rate of convergence to equilibrium, that means the mixing rate of the Markov chain, is determined by the second largest eigenvalue modulus of T, abbreviated SLAM. You can see here an example of small Markov chain, its transition matrix T, and the eigenvalues of this matrix are lambda 1 and lambda 2. Because the lambda 2 is the second largest eigenvalue modulus, has a second largest eigenvalues modulus, the mixing rate of this Markov chain is 0.1. Fastest mixing Markov chains. The problem here is to find the edge transition probabilities that give fastest mixing Markov chains. That means that minimize SLAM. This problem can be formulated as a convex optimization problem, which can in turn be expressed as a semi-definite program. That means to minimize SLAM subject to P higher than 0, P1, here 1 is unary matrix is 1, and P equals transpose of P. For the analysis of a data flow graph, one has to transform it to the corresponding Markov chain, then to use this algorithm, that means to perform semi-definite programming, and then revert back to the optimized Markov chain, that means to the data flow graph. Also, we have a slowest mixing Markov chains, and here the problem is opposite to the fastest mixing Markov chain. That means to find the edge transition probabilities that give slowest mixing Markov chains, that means that maximize the SLAM. As the algorithm for finding slowest mixing Markov chains is not known yet, this problem cannot be solved with semi-definite programming. However, it can be solved by examining all possible chains and finding one with the highest SLAM. That means with the slowest mixing speed. As Vini is analyzing metabolic processes, which are deterministic in their nature, Markov chain must be replaced with data flow graphs. All nodes and edges of data flow graphs must have, of course, some numeric value. Numeric values in Vini's data flow graphs represent energy values. For nodes, these are free binding energies between receptors and ligands. For edges, these are interactions among receptors. Here, the data flow presentation of Vini's search for better drug. Starting with the first drug, Vini gets somatic expressions and mutations from cosmic database, then drug and receptor structures from protein data bank and from DrugBank, cancer pathway from KEGG, and then it constructs data flow graph and data flow matrix 1 and computes SLAM 1. The process is repeated for the second and for all drugs or chemical compounds under the investigation. At the end, Vini produces a list of SLAM values, sorts them in an ascended order, and the drug or chemical compound with the highest SLAM will point to the probably good candidate, and with the lowest to the not so good, say we, drug or compound candidate. So let's take a small walk from the pathway to SLAM. In the first step, pathway receptors are replaced with their structures. That means with their three-dimensional structures taken from protein data bank. Or in case these structures doesn't exist in protein data bank, Vini uses tools for predicting their structures. In step 2, ligand, which can be either an approved drug or chemical compound under investigation, is docked on receptors and free binding energies are computed. Data flow graph is created with nodes representing free binding energies and edges representing interactions between receptors. In the last step, graph is presented with a matrix and its SLAM computed. Now we really need a little break from SLAM, Markov chains, mixing rate, data flow graphs, etc. Here we can see schematic presentation of Facebook friends, and approximately Facebook user has about more than 200 Facebook friends. But the number of real friends is considerably smaller. In my case, I have only few friends, and among them are Velika and Mala Vini, produced big and small Vini, and that is the reason why I named this in silico model of cancer Vini. Let's take a short look at Vini components. First, their application interfaces to external databases and portals. Of course, one very important database is DrugBank, and Vini acquires drug structures from DrugBank. Then KEGG, from which Vini acquires pathways of diseases. Then UniProt. UniProt is contacted in case no structure, that means three-dimensional structure, is available. Then Vini contacts UniProt database, acquires from it FASTA sequence of protein, and converts FASTA to 3D structure with special tools, programs that we will see next. Then also one of the most important components is protein data bank with its more than 100,000 protein structures. And because the KEGG pathways, the most components are genes and proteins, then Vini frequently contacts protein data bank for these structures. From Cosmic, Vini acquires genetic expressions and mutations for various types of cancer, and it uses a Swiss Institute of Bioinformatics portal when it needs to predict some protein structure. And then PubChem. PubChem, in case a ligand, that means a compound under investigation is not an approved drug, then it acquires its structure from a PubChem database with hundreds of millions of chemical compounds. Take a look at the third-party programs. Of course, the most important in Vini, this is Autodoc Vina, which performs docking and free binding energy computing between proteins and small molecules. MGL tools, which are used for preparing receptors and ligands, and both these tools are from Scripps Institute. Then Rosetta Doc and Rosetta Antibody, Vini uses to perform docking between two proteins and to predict structure of monoclonal antibody drugs. UCF's Chimera is used for preparing finally protein structures. OpenBubble to convert chemical files from one format to another, and AlphaFold to predict receptor protein structures. Then also Reduce, a famous program from Duke University, which Vini uses to add hydrogens to proteins. Let's take a small look at use cases. First, what is the accuracy of the Vini model against the clinical trials? We validated that. So we let Vini to process 70 cake cancer pathways against 132 cancer drugs. Evaluation of the results was performed by taking for each cancer pathway 5 drugs with the highest SLAM values and then examining their efficiency from the available in vivo studies. In the right table, you can see hits, that means drug is drug efficient, are labeled in green. Misses, that means drug is not efficient, in violet. And yellow means studies exist, but results for some reasons are not available. White field means in vivo study was not performed for that cancer type and drug. The probability that Vini will correctly predict the efficiency of a drug against a specific type of cancer is defined as the ratio of the number of green fields to the sum of green and violet fields and is 0.793. Next use case, the accuracy of Vini in predicting the efficacy of cancer drug combinations. We let Vini calculate the most effective 2-drug therapies against non-small cell lung cancer, small cell lung cancer, and pancreatic cancer. Vini calculated the combination of vinorelbine with paclitaxel as the most effective against non-small cell lung cancer, then the combination of everolimus with doxorubicin as the most effective against small cell lung cancer, and the combination of everolimus with paclitaxel as the most effective against pancreatic cancer. The results from existing clinical studies strongly confirm the results Vini predicted. The next use case is the accuracy of Vini validated against NCI 60 GI 50 data. So Vini computed SLAM for 50 FDA-approved cancer drugs and then over 30 NCI 60 cell cancer lines. Then it correlated computed SLAM values with GI 50 data for these lines. Pearson correlation values have shown good values ranging from 0.40 up to 0.64. Also to mention, GI50 is the concentration that results in inhibiting cell growth by 50%. And this is an example of negative Pearson correlation, where two sets correlate inversely with each other. The higher SLAM is, the lower the concentration of drug is needed. And then the case 4, we deployed Vini against COVID virus. Vini was applied at the very beginning of the outbreak of COVID pandemic to find the most promising triple combination of existing antiviral drugs against this virus. Structures of 50 FDA-approved antiviral drugs were downloaded from DrugBank and used in this experiment. The combination of HIV drugs ripivirine, cobicistat, and abacavir was identified as the most promising candidate for blocking spike glycoprotein activity. Vini is the next generation supercomputing application. It was powered on the Mare Nostrum supercomputer in Barcelona to enable the creation of an advanced protein-protein artificial intelligent docking model based on neural networks. Currently, Vini is under further development on 3 other supercomputing facilities: Vega in Slovenia, Bura at Rijeka University, and Isabella Supercluster at Zagreb University. Also, we need to note that Vini is an open source application. However, third-party programs and databases are licenses, and they are for free for academic use. So if you find Vini silico model interesting, then you are more than welcome to join our efforts. What is the future of Vini research? Towards the integration of NAM-driven coarse-grained molecular dynamic protocols. And because now Vini is working on all atom scale, and simulated time periods are in the range of nanoseconds. When we move to the coarse-grained molecular dynamic, then we will be able to hit more cancer phenomena than just now, because then we will be able to work in milliseconds range. Here you can see some of Vini friends, and these are researchers that take a look at Vini, published some papers related to Vini, were involved in the preparation of project proposals, provide some good or even brilliant ideas for the further development of Vini. And I hope this number of friends will grow to the more than it is now. So I would like to thank you for attending, and now you are free to ask the questions.